Viruses exhibit a remarkable diversity, infecting organisms from every domain of life and thriving in virtually all ecosystems. But how did this incredible diversity arise, and what are the origins of viruses?
The virus first hypothesis suggests that viruses could have been the original life forms, emerging independently in the prebiotic world. Imagine a primordial soup, rich with organic molecules like RNA and proteins, all freely interacting in a chaotic dance of chemistry. In this environment, the theory proposes that viruses began as self-replicating entities, tying into the well-known RNA world hypothesis. This theory suggests that life originated within RNA molecules, which acted as both genetic material and catalysts for chemical reactions. These early RNA strands could replicate and mutate, competing for scarce resources. Some even gained a competitive edge by parasitizing primitive lipid vesicles, tiny bubbles that concentrated biomolecules. Over time, protective protein shells formed around these RNA strands, evolving them into the first viruses. However, a key question remains. If viruses existed before cells, how do we explain their reliance on host organisms? A defining feature of a virus is its dependency on a host cell for replication. Without cellular life, how could they persist, given their lack of machinery for independent metabolism or reproduction? This uncertainty about the reliance on host cells leads us to alternative explanations, such as the one detailed by Hypothesis 2.
The regressive hypothesis is the idea that viruses evolved from more complex cells that lost certain functions. Some virologists have hypothesized that autonomous individuals initially developed a symbiotic relationship. Over time, this relationship turned parasitic as one organism became more and more dependent on the other. The once free-living parasite then lost previously essential genes, eventually making it unable to replicate independently, becoming a virus. Mimivirus is a virus that supports this hypothesis. It is much larger and more complex than other viruses, alluding to more complex ancestors. Additionally, it contains a large repertoire of putative genes associated with translation. Such genes may be remnant of a previously functional translation system.
We've explored the idea of viruses coming from complex origins, but a third theory shifts focus to a simpler, more gradual path of viral evolution.
Third hypothesis, progressive hypothesis, suggests that viruses did not originate from more complex cells, but instead evolved from genetic material that escaped cellular genomes after the first cells appeared, becoming independent infectious agents. This process has started from unequal cell division, producing two mini cells containing chromosomes A and B, both lacking full protein synthesis machinery. Chromosome A is destroyed, while chromosome B survived by associating with a protein coat, gaining the ability to infect other cells and becoming a virus. Retroviruses like HIV support this idea as they are closely related to retrotransponons, mobile genetic elements that made up about 42% of the human genome. This connection undergoes the gradual evolution of viruses from cellular components.
While their origins remain uncertain, most agree that viruses likely evolved through multiple mechanisms, each suited to different viral lineages, as follows.
For mutations in RNA viruses, genetic diversity results from insertion, deletion, and substitution. The rate of occurrence and the fitness effect define how much impact these mutational pressures have on the evolution of viral populations. All mutations interact and accelerate the host virus arms race and produce the variety of viruses and viral proteins we observe today. Use SARS-CoV-2 variants as an example.
Reassortment of viral DNA is a mechanism of genetic exchange that occurs in segmented viruses. It involves the mixing of genomic segments when two related viruses infect the same host cell. The genomes from each virus are replicated, followed by the assembly of new virons, in which genome segments from both parental viruses randomly mix. The newly formed virons carry hybrid genomes that are genetically distinct from both parental viruses. A famous example of this is the reassortment between human, avian, and swine influenza strains, leading to major pandemics such as the 2009 swine flu.
In conclusion, the origins of viruses remains uncertain, and further research is needed to reach a definitive answer. One leading modern theory, the chimeric model, blends previous hypotheses in an effort to solve this puzzle. Regardless, viruses continue to evolve and thrive today, demonstrating their remarkable ability to exploit a diverse range of hosts and environments.